Today we can see the structure of DNA. So what is DNA? DNA is deoxyribonucleic acid and it is the hereditary material and it will be present inside the chromosome. So inside the chromosome gene will be there and inside the gene this DNA will be present. And this DNA contains the genetic information. And this DNA is double helical structure, that is the spiral structure, true spirally coiled structure. And its structure was proposed by James Watson and Francis Crick in the year 1953. Rosalind Franklin and Maurice Wilkins, they obtained the structure of DNA by X-ray diffraction studies. X-ray diffractor is an instrument which is used to study the structure of minute particles. Based on their studies, this James Watson and Francis Crick, they proposed the 3D model of DNA. And because of that, Watson, Crick and Wilkins, they got Nobel Prize in the year 1962. Right. Then we can see the composition of DNA. So what are the chemicals which are present inside the DNA? This DNA is made of millions of nucleotides. So as it is containing more nucleotides. So if the number is above 3, we will call as poly. Hence this is also known as polynucleotide. So what are the constituents of this nucleotide? So nucleotide means it contains a sugar molecule, nitrogenous base and the phosphate group. So we can write like this: nucleotide is equal to a sugar molecule plus nitrogenous base plus the phosphate group. And the sugar molecule which is present in this is deoxyribose sugar, and the nitrogenous bases are, there are two types of nitrogenous bases. They are the purines and pyrimidines. The purines are adenine and guanine and the pyrimidines are cytosine and thymine. Then the one phosphate group. So these all together called as the nucleotide. And here the sugar molecule and the nitrogenous bases, these both we can call as the nucleoside. So nucleoside is equal to a sugar molecule plus nitrogenous base. And nucleotide is equal to a sugar molecule plus nitrogen base plus phosphate group. Also we can call this as the nucleoside plus phosphate group. Can you understand this? This nucleotide is equal to a sugar molecule plus nitrogenous base plus phosphate group, or it is also called as the nucleoside plus the phosphate group. These are the chemical composition of DNA. Next we can see what this Watson and Crick says about the DNA, how they described this DNA. So they said the DNA molecule consists of two polynucleotide chains. So these are the true polynucleotide chains. So just for your understanding, I am making this picture. You can see this picture. You can use this picture for your exams, and you can take this picture for your understanding. So two polynucleotide chains, and these chains are double helix structure, that is they both are spirally coiled structure, and these both chains will run anti-parallel to each other, that is one will run from here to here and the another will from here to here, that is 5 prime to 3 prime, then this one from 3 prime to 5 prime. So these all you will study in later classes. So just you can know that one will run from here to here and another from here to here, that is they both are anti-parallel to each other. And we know that this DNA contains a sugar molecule, nitrogenous base and phosphate group, right? In that, this nitrogenous bases will be present at the center. So what are the nitrogenous bases? The purines and pyrimidines. They are the adenine, guanine, cytosine and thymine. These are the nitrogenous bases and these will present at the center. That is in between these two chains, these nitrogenous bases will be present. So here this nitrogenous base, that is A, T, G, C, A, T, G, C, A, T, G, C. Like that this nitrogenous bases will be present. And these nitrogenous bases are linked by the sugar and phosphate group. So the sugar and phosphate group. So in this we can see remaining nitrogenous bases will be present at the center, and the sugar molecule and the phosphate group is remaining, and these two molecules will be present in this chain. So here the sugar molecule and the phosphate groups are present, and this sugar molecule and the phosphate groups, they are the backbone of our DNA. So the backbone of the DNA is the sugar and phosphate group. And here the adenine will always pair with the thymine, and the guanine will always pair with the cytosine. That is, the purines will always pair with the pyrimidines, and the pyrimidines will always pair with the purines, and the purines itself they will not pair, and the pyrimidines will not pair with the pyrimidines. Always the purines will pair with the pyrimidine. So adenine is the purine. It will pair with thymine. So the adenine will pair with thymine with two hydrogen bonds. So these are the two bonds, and this will pair with thymine. And the thymine is the pyrimidine. This thymine will always pair with adenine, that is with two hydrogen bond. A will come here. Then guanine is the purine. This guanine will always pair with cytosine with how many bonds? With three hydrogen bonds. Three hydrogen bonds will be present. And this cytosine will pair with guanine. Guanine with three hydrogen bond. Now it's your turn, you have to say. So adenine will always pair with what? Adenine will pair with thymine. So adenine will pair with thymine. With how many bonds? With how many hydrogen bonds? With two hydrogen bonds. So we have to put 2 here. And then thymine. Will always pair with adenine. So here we have to put A. So with how many bonds? With how many hydrogen bonds? With 2 hydrogen bonds. And this guanine will always pair with cytosine. Guanine will always pair with cytosine with how many bonds? With 3 hydrogen bonds. So like that, this cytosine will pair with guanine with 3 hydrogen bonds. Like that, this bonding will be there. And these hydrogen bonds will make the DNA molecule stable. The DNA molecules are stable because of these hydrogen bonds. And like this, these strands, here these base pairs are there, these bases are there, and by these bases only this strand was filled. In this strand, this bases was filled by according to this bases. Hence, this strand, these base pairs are called as complementary base pairing. Complementary means filling. That is, this strand got filled because of this strand. That's why this base pairing is known as complementary base pairing. Then each turn of the double helix is 34 angstrom. That means here that is 3.4 nanometer. That means from here this point to this point. That place only we will call as one turn, and in this turn's length is 34 angstrom. It was also known as 3.4 nanometer, and there will be 10 base pairs will be present in one complete turn. So one turn contains 10 base pairs. So here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So these 10 base pairs will form one complete turn. And the nucleotides in a helix here are joined together by phosphodiester bonds. So the bases are joined by the hydrogen bonds, and here the sugar and phosphate groups are at the chains, and these are linked together by the phosphodiester bond, and this phosphodiester bond will be present at the chains, and the hydrogen bonds will be present at the center. And this Chargaff's rule, what this Chargaff is saying is, Erwin Chargaff, he states that, so inside the DNA, this adenine and thymine, this guanine and cytosine, these all are present, right? So here he is saying that the proportion of adenine will be always equal to that of the thymine. That is, if there is 10 adenine is present means there will be always 10 thymine, because adenine will always pair with thymine. So if there is 5 adenine means there should be 5 thymine. And the proportion of guanine will be always similar to that of the cytosine. So if there is 15 guanine means this 15 guanines will always pair with the cytosine. So there should be 15 cytosine. And if there is 25 guanine means there should be 25 cytosine. So these are all about the structure of DNA.